Okay, hello, welcome to this video from Mr. Gill Science Stuff on the polymerase chain reaction. This is the first video, real lesson video on the Biotech Basics playlist. So it's as good a place to start as any in the biotech topic. It is on most A-level syllabuses, but as I said in the introduction, all these topics do interlink. They are interrelated, and if there's anything that you don't understand in this, it might be because you haven't covered that bit yet. So watch some of the other videos, and if you still don't understand, get in touch and ask. Okay, so what is the polymerase chain reaction? Well, essentially it's a means of taking a very tiny sample of DNA, can even be one single molecule of DNA, and amplifying it to make a very big one. That's the purpose, and it is absolutely standard lab practice. It is a real basic in your biotechnology toolkit. It's quick, it produces masses of cloned DNA, it's easy, it's cheap, and it's really effective. Essentially, it's DNA replication in a test tube. So I'm assuming you understand semi-conservative replication and how DNA replicates within a cell. This is essentially doing the same process but in a test tube. Okay, it was invented in 1983 by a guy called Kary Mullis, who won the Nobel Prize for that in 1993, which makes the polymerase chain reaction exactly the same age as me, which is wonderful. Right, so what kind of uses are we talking about? Well, forensics, diagnostics, selection or amplifications of any sort of genes, looking for individual sections of DNA and their relatedness, and many more. If you can think of an application where you might need more of a particular strand of DNA, then the PCR reaction is what would be used as a standard lab process. And I've given a list of examples down there, but there are as many as you can think of. And quite popular at the moment, quite familiar you should be with the idea that we're looking for tests for the COVID-19 virus, for the coronavirus, and that test that's looking for the presence of the virus is based on polymerase chain reaction, right? But that's for another video. So what do you need? Well, this is basically the same ingredients as you would need for semi-conservative replication. You need the basic building blocks of DNA, your adenine, thymine, cytosine, guanine. You need a template to copy, some sort of sample that you're trying to copy. You need an enzyme, DNA polymerase, which is the enzyme that makes a polymer of DNA, so nice and simple. A new ingredient you probably won't have heard of before, called primers, which we'll get to in a moment, and a heat cycler, which is literally a box like the one pictured, but which just gets hot and cold and hot and cold and hot and cold. Okay, and that is a programmable heat cycler, and they are absolutely ubiquitous pieces of lab equipment. You can pick them up on eBay. I was just having a look before recording this video for under 100 quid. Right, they are absolutely ubiquitous, dead common. Right, you find them in every university, hospital, and most school labs nowadays. So, in short, this is the short version of what happens in a PCR reaction. Right, first thing, you take your length of DNA you want to amplify, you mix it with the 4 nucleotides, A, T, C, and G, and you add the enzyme DNA polymerase. Okay, you have a cycling protocol of different temperatures, heating up, cooling down, heating up to precise temperatures depending on your protocol, and the DNA will then be replicated many times. Okay, now that obviously is a very short version. Right, if you look inside a heat cycler, inside the heat block while it's working, you would see something like this, where you've got tons and tons of tubes in one heat block being heated up, cooled down, heated up, cooled down. As I say, this equipment is so common, I have actually known university academics taking old heat cyclers and literally setting them to a nice even 70 degrees or so, and having them in their office to put their cup of tea on. Right, they're that common. So let's look at that in a little more detail about what's actually going on inside these heat cyclers. Well, initially you've got your original DNA there, which is our double helix you should be very familiar with. Okay, so this is our double helix. First thing we want to do with it when it's mixed with our ingredients, right, and in reality, I know it says add primers down there, in reality all this stuff would be added at the beginning into one tube, and then you just leave the thing running. You heat this thing up to 95 degrees C, and that's called the initial denaturation. It breaks the hydrogen bonds between the 2 strands of DNA and separates those into 2 single strands. Now usually in the first cycle that heating stage is a little bit longer than it is in subsequent cycles, because remember you've got to separate out the entire strand of original DNA, the entire double helix. That's quite a lot of hydrogen bonds. Now in nature you should know that usually that would be DNA helicase that does that in semi-conservative replication. Here we're using heat. So we end up with 2 single-stranded pieces of DNA. So first thing we do, initial denaturation, you separate the strands by heating it to 95 degrees C, 2 minutes, depends on your protocol as to how much DNA you're trying to separate and what precisely that you need to do. Okay, then next, right, our primers come into action. Right, these are the new thing you probably won't have heard of if you haven't done this before. So you separate the strands out like this, and now you've got some free hydrogen bonds. Now what primers are tiny little lengths, 20 base pairs long, something like that, or 20 bases long, called primers, and they essentially sit on the end and they give the DNA polymerase somewhere to start. They give a starting block. Now, I always like to liken biotechnology stuff to children's toys. This is very much like Hot Wheels. You've ever had Hot Wheels with a little car launcher, right, where the car can be put in and pushed down the track. This is basically the starting block like that, but for DNA polymerase. Now, of course, this means you have to know what sequence you're looking for. You have to know what sequence you want, and then you have your primers manufactured ahead of time. So you might be thinking, well, how on earth do we know what the sequence should be? Well, that's for another video, okay, how we initially find those initial sequences. But let's assume this is a sequence that you're looking for, that you know what you're looking for, you know what you're amplifying, and you can get hold of the primers fairly easily. Okay, and that's what's going on in viral diagnostics at the moment. Right, we know some of the sequences we're looking for, so therefore the primers are quite readily available. So you add the primers, you cool them down to 40 degrees C, right, to let the primers form their hydrogen bonds, and then you heat it up to 72 degrees C, and the DNA polymerase starts to make those phosphodiester bonds, right, join those bases on, one after the other, and you get your 2 double helixes from 1. Okay, so you cool the primer, cool to 40 degrees to allow the primers to anneal. That's complementary base pairing. Lift it up to 72 degrees, and we'll get to the reason why it's 72 degrees in a moment, and that will manufacture your 2 double helixes from 1. Then, from point 4 down here, you simply go back round to point 2. You heat them up again, so they separate, you cool them down, so more primers anneal, and from 2 you go to 4, then from 4 to 8, 8 to 16, and so on and so forth. Okay, so very rapid amplification. So let's look a little more detail at what happens in one particular strand. Okay, so first thing, your initial denaturation at 95 degrees C separates the strand. Your primers will bind onto whichever stretch of DNA it is that you're looking for. Okay, so it could be a tiny little section out of a huge initial molecule, but they will bind on those places. Now, usually you have them so that they nicely mark out a particular section of DNA that you want to amplify. Okay, so then, right, we need to talk about why it's 72 degrees, why we lift this up to 72 degrees C. That is because you've got a problem here, right? DNA polymerase extends the primers and completes the replication of the rest of the DNA. But if every cycle you have to heat it up to 95 degrees C to break the hydrogen bonds, what do you think you're going to do to the polymerase? Well, if it's human normal polymerase, then you will denature it, and you'll have to add a new enzyme every single cycle. So we don't do that. We use a special form of DNA polymerase that comes from a bacteria that was found to live in hot springs in Iceland. Okay, it's called Thermos Aquaticus, because biologists are terrible at being imaginative but do know a bit of Latin. So Thermos Aquaticus, yeah, hot water-based living thing, and its version of DNA polymerase has an optimum temperature of 72 degrees. So surprisingly enough, that's the temperature that you work your PCR machine at. Okay, and so it's not denatured when you heat this thing up to sort of 90, 92 degrees. So that's Thermos Aquaticus, and Thermos Aquaticus DNA polymerase is often seen written as TAC DNA pol, right? And that's what that abbreviation there means. It's something you should know, right? That we're using this special enzyme so it doesn't denature. Right, onwards. So there's our guy here, like in one of those hot springs, looking for the natural habitat of where Thermos Aquaticus would live in a completely unstaged and naturalistic photo. Right, that's the structure. You can see the green is the backbones of the DNA moving through the blue, which you should recognize as alpha helices of the TAC DNA polymerase. Right, so then once we've got that enzyme in there, we get extension. So at 72 degrees C, we get a lovely bit of extension there, so we get manufacture of our new strands, right, and that gives us our amplified bit of DNA. So from that point, we then go back to the start. We heat it up, 95 degrees C, we separate out the DNA strands, we cool it down to 40, and we heat it up to 72, and then again our primers anneal, and we get our extension. So as you can see, look, we've now got 1, 2, 3, 4 double helixes from that initial 1, and that's after generation 2. Okay, so it doesn't take a genius to work out this is an exponential growth now. So you've got initially 1 molecule, then 2, then 4, then 8, then 16. After 10 cycles, you've got 1024, and then after 20 cycles, you've got over 1 million copies of that particular bit of DNA, and that is a very useful amount of DNA to be able to work with for whatever it is that you're doing afterwards. Forensics, diagnosing a particular illness, looking to do some genetic engineering, so you've got lots of copies of that particular strand of DNA that you want to put into something else, all sorts of different things. So it's completely automated. A few hours, you get a tiny sample of DNA is amplified millions and millions of times, right? And the little bit of maths that you could be expected to do is this same thing as 2 to the n, right? The idea is sort of using n as the number of generations, number of PCR cycles. You started with 2, right? And then you use 2 to the n to work out how many copies of that DNA you end up after a certain number of cycles. It's exactly the same bit of maths you may remember you had to use when calculating the number of possible gametic combinations in when you did meiosis in year 12. Okay, so 2 to the n, simple using exponentials, not tricky. Obviously, there's an important notice here. You might have noticed that when I talked about forensics at the beginning, the forensic scientists were dressed in, the big suits with the little boots on and the gloves and all kinds of things. Any contamination of the DNA will also be amplified. So the forensic scientist gets a skin cell into the sample, and that will contaminate the sample, and that could be amplified as well. So obviously it's become more and more and more important that people looking into crime scene research do not contaminate the sample, and we're going to talk more in another video about how we avoid that in the lab. Okay, that obviously, if it's in forensics, right, if in crime, it can cause problems in court cases because people can argue that this was a contaminated sample and therefore doesn't recommend that you can't convict someone on the basis of it. So that's how the PCR reaction works, right? It's literally a DNA photocopier. Right? Final note, it allows you to do other variants, which we'll come to in other videos. So it allows mixing in other versions of ingredients. So if you want to be able to detect your DNA afterwards, right, you can put in radioactive or fluorescent molecules, and that is one very quick and easy method of doing diagnosis, and it's one of the ones being used for detecting coronavirus, is to use radioactive or fluorescent molecules in there that if that particular strand you're looking for is there, and you start to manufacture these new molecules, that they glow or they develop a photographic film because they're radioactive. So that's that, using those as a marker. And then we're going to come to, when we talk about sequencing, these so-called double deoxynucleotides, but we'll learn about that in the video on sequencing. So they're important in other processes. And that, as far as A-level standard goes, is what you need to know about PCR. However, there is always a few people in every class, there are always a few smart cookies who ask the obvious question, which I'm assuming you've all thought of, the obvious omission that I've left out. We need to go back a few slides to this. Okay, when I showed you the primers annealing and put those 2 markers that's our target bit of DNA, and then did this, okay, the obvious question is, how does the strand know when to stop? Why? So it's coming from the 5' end of the primer here down towards the 3' end, and why does the polymerase suddenly magically know to stop there? Why does it suddenly magically know to stop there when actually, there's no relationship? These two are separated. So primer binds here, polymerase starts to build the strand, it gets to this point here. Why does it not keep copying further on, right? And the simple answer to that question is it doesn't know. And in the first generation, it will indeed continue to copy a huge swathe of the DNA. It will just carry on and on and on. So you'll get your initial target bit of DNA here, but you will also get a big long tail, right? Now, to understand why you only end up with that particular chunk of DNA, you have to go into the next generation, because in the next generation, when these things get split up again and the primers anneal and your extension happens, right? Because the strands are anti-parallel, so in each generation you're going to be going in a different direction, right? As you can see up here, this strand that's being formed here does reach a hard end. It reaches the original primer, and then the DNA polymerase doesn't have anything to copy, so it literally just drops off, right? The same is true down here, yeah, where it literally just drops off. Okay, now yes, of course, in every generation you're going to get that one original, because that original strand of DNA, the black stuff in this instance, the original sample is still there. So in every single generation you're going to get another molecule of these long molecules, another copy of these long molecules, but that's only one per generation. So you get one in the first generation, you get two, then three, then four. Whereas these ones, okay, the ones that are the right length, amplify exponentially. So they go from 2, 4, 8, 16, 32, and they amplify so much faster that essentially the long ones become a negligible contaminant after the first few generations. Okay, so it's those ones there, right, that end up amplifying exponentially after the first generation. So if you spotted that question, well done, right? Good job. That's a high-level question, higher level than you will need to talk about at A level, right? But there's always a percentage of people in every class who go, hang on a minute, how does it know where to stop? And that's the answer. Okay, so next video on Biotech Basics will be gel electrophoresis, and that will go up as soon as I have written and recorded it. Many thanks. See you next time.